All right, here we've got our video for section 17.7 on mutations, and we were specifically looking at small mutations within a genetic code. Earlier in the year we had looked at some chromosomal mutations. I forget which chapter that was, but anyway, looking at small mutations, start out with an example here of sickle cell disease, and sickle cell disease is in the hemoglobin gene in blood, and if you remember, hemoglobin is what carries oxygen in your blood. And so here's just a segment of a normal hemoglobin gene, or the mRNA, or the DNA, the normal gene. People with sickle cell disease, they've got one letter difference in a very, very large gene, actually, where there's a T here in the middle of this codon, and it's actually an A for someone who has sickle cell disease. Well, in the normal hemoglobin for someone without sickle cell disease, if this is the triplet in the DNA, the CTT is the triplet in the DNA, then our codon is GAA, because that's complementary there, GAA in the mRNA, and then that codes for GLU, which is glutamine, I believe. Anyway, that's the normal. Well, when we change this this T to an A, which makes a U in the middle here, that all of a sudden makes valine, and it is just one amino acid in a really big protein, but that one amino acid change happens to be at the an active site within the hemoglobin, and so it's not as effective at what it's supposed to do, and it causes a whole bunch of problems for people who have sickle cell disease. So that's just an example. This type of mutation where we just change one letter, that's called a point mutation, and we're going to take a look at a couple of different types of point mutations here. So here's our diagram, or a picture of different point mutation possibilities. And so we have our wild type. We're actually going to look at the mRNA rather than tracking all the way back to the DNA. But if a mutation, here's our wild type mRNA, and we're going to just look at a couple different spots along here where there could be mutations and what they would mean. The first type of mutation is this base pair substitution mutation, and that's what we saw in the sickle cell disease, where you just have one letter replaced with another. So the C that's right here in the mRNA is replaced with a U, and again, this mutation would have happened back at the DNA, and because the DNA is different, then that's going to make the RNA different, and then that's going to make the protein different potentially. Okay. In the case of sickle cell disease, it did make a difference in the protein. In this case, though, it ends up that it is the third part, or the third nucleotide in the codon, so it's GGC. I changed it to GGU. I don't want the light in the classroom, and actually it's alright with the light off, so we're going to leave it that way. The GGU ends up coding for the exact same amino acid, so glycine in both cases. So this is a genetic mutation because we changed the DNA, but this one doesn't actually change the protein. Okay. When it doesn't change the protein, it is called a silent mutation. So the mutation does exist, it's just you have no way of knowing it because the protein doesn't actually change. And again, that where we replace one letter with another, that's called a base pair substitution here. Base pair substitution. Okay. There's other results that can happen within this base pair substitution situation. Anytime there's a mutation, it can either lead to, right here we have missense and we have nonsense. Okay. Missense means that I changed the letter, so here we're going to put an A in this spot instead of the G that was originally there. And that A is actually changing the amino acid. So what used to be glycine up at the beginning is now serine down here. So we changed what the amino acid is. So technically this codon, AGU, it still makes sense. It's just the wrong sense. So they say it's missense is what the mutation is. This type of thing, changing one amino acid, depends what the new amino acid is compared to the old amino acid. If serine and glycine are similar, then we won't see a huge change in the protein in the end. If these two are very different, like one's acidic and one's basic, or the positive and negative charges are different between serine and glycine, all of a sudden the structure of the protein is going to be different, meaning its function is going to work a little bit differently. It may work, the protein may do its job, maybe it doesn't do its job, but there's a definite change happening here. Okay, so that's missense. The other option, if we look here, if I go all the way up to the top, the AAG is supposed to be lysine. If I change it to UAG, that ends up being a stop codon. Anytime that you hit a stop codon relatively early on in the protein, we're going to call that a nonsense mutation, okay, where it makes a stop codon. If you have a stop codon, it's almost always going to render this protein useless. So you have a non-functional protein. Okay, so that's the nonsense, missense, and silent mutations, and all of these were cases of base pair substitutions. Okay, now there's some other minor, what we call point mutations, just a mutation at one spot in the DNA that give us some different results. Results have some different names, so we'll look at those now. All right, these next two types of mutations, they are also they are also point mutations, but we call these mutations that shift frames. Okay, and what they are is they're insertion, and let me get my pointer here. They're insertion and deletion mutations, and what they do is they call something called a frame shift. So insertions and deletions are type of point mutations. They are also type of frame shift mutations. So let's look at an insertion. So here, the normal protein or the more normal code has AUG followed by an AAG, and what we're doing is we're going to add an extra U in the middle there. So we have AUG, add an extra U, and then this is our AAG. Okay. Well, in this situation, when we talk about the frame shift that's happening, our codons are actually going to change. So if I draw in where our codons are on the original, I've got AUG as a codon, and then AAG, and then UUU, and then so on. Okay, we go through this. If I look at this new sequence here, still have AUG as my first codon, but now my second codon is UAA. And my next is GUU, and then UGG. And if I compare these, I took my UUU, and it's now a part of two different codons down here. The UUU that was up there is part of two different codons down here. And the GGC that's up here as a codon, it's part of two different codons down here. So the reading frames, they call it, but the groups of three have shifted because we inserted this U into the middle. Okay. In this case, this led to an immediate stop codon, and so this is a nonsense mutation, okay, because it's not making any sense. Sometimes you can do a frame shift mutation like the one down here, where we're going to do a frame shift deletion, where there's supposed to be a UUU, and we're going to remove this U. Okay, again, there's going to be a frame shift that happens. We have our normal AUG for our first, AAG for our second, that's good. But now instead of UUU, we have UUG, and then everything from this point on is going to be screwed up. Okay. Well, this, if from this point on, where the mutation happened, this is a missense thing here, right? Because it's still coding for an amino acid, it's just a different amino acid. And this is coding for an amino acid, it's just different, and so on and so on. At the pause. All right, I got interrupted there. Somebody walked into the classroom, but I kind of forget where I was at, but I do know I was talking about this scenario right here. And this is, again, a frame shift causing a lot of missense. Okay, so there's still going to be some kind of protein made, but almost guaranteed it's not going to work or do what it's supposed to do. So we can have individual insertions or deletions, or we can actually have insertions and deletions of a complete codon. So here we have an insertion or deletion of 3 nucleotides. Technically, there's no frame shift on this one, because we're going to keep, everything's going to stay in this, the U here is going to stay together, the GGC, because we've just added a codon. Okay, you can also delete a codon, depending, I guess this is a deletion here. Okay, so those are insertions and deletions, and we get these point mutations. And a couple things to add into the discussion here. One is, for the most part, none of this matters most of the time. Okay, most of the time it doesn't matter if I have an insertion or a deletion or anything like that in every single cell in my body. If it were to happen in one of those at a time, it doesn't usually matter, because maybe it makes a protein not work. But who cares? I've got, let's say I actually had the mutation in one of my blood cells that gave that cell sickle cell disease. Okay, it wouldn't matter. I've got a whole lot more blood cells that work the right way. That one mutated cell is not going to be a big deal. Okay. Where it would be a big deal is in the production of gametes. Okay, and so if there's if that mutation happens in the production of a gamete and gets passed on to the next generation, all of a sudden sickle cell disease, that allele, would have entered the population, or would have entered my ancestry if it was me. Okay, so that's when it matters most, is if it's going to be an insertion or deletion or anything that leads to mutation, and it's happening in one of the gametes. Okay, because then the next generation will be carrying it. In the time that it does matter for your non-gamete, so all your diploid cells, the times that it would matter is if it's going to lead to something like cancer. okay, which can be like a, well, we've talked about cancer earlier, so if it leads to like a cancer type thing. Last thing, the book mentions something called mutagens. Mutagens, I'm going to run out of room. A mutagen is some sort of chemical, that's horrible, some sort of chemical that increases the chances of mutations happening, or a physical or chemical thing. And so they list a couple of different types of things. Like they know that X-rays and ultraviolet light and things like that can cause mutations to happen, not just increase the chances of, but actually cause the mutations to happen. They also know there's certain types of chemicals that when introduced into the body, they can attach to the DNA or attach to a nucleotide or do something like that, and end up causing mutations. They call them mutagens, or sometimes they call them carcinogens, because things that are mutagens, the only time you'd ever notice them is if they caused cancer, and a carcinogen is something that's a cancer-causing agent. So if you ever hear that word, carcinogen. But anyway, so there are, and there's actually a huge study, area of study and studying the different types of mutagens and carcinogens that are out there. So I think that's it. Oh no, you know what? I'm going to show you one more slide here. Just showing you this picture. This is the picture that is the overview of the entire chapter here. So I would encourage you, pause the video or go into your book, find this figure. It's at the very end of the chapter. Make sense of it all. Okay. It even has transcription from chapter 16 involved in there as well. So we'll end it at that. Hopefully this chapter goes well for you, and enjoy the test as it comes up.